second part of the module now, which is very much devoted to thinking about these molecules called therapeutic biologicals. I appreciate in your group work, some of you will already have studied some of the things that I'm going to be talking about. The reason we focus on this topic in this course is because of their immense importance to the modern pharmaceutical industry, which is an industry that I know some of you are interested in finding careers in the future. And I want to kick off just by answering this question about what's the world's biggest selling drug in terms of revenue. These are slightly old figures. These figures come from a couple of years ago. The pharmaceutical industry tends to guard these figures rather jealously. So figures for the last couple of years are hidden behind paywalls, and you have to pay large amounts of money to get them. But these are the figures from 2015. And this may really surprise you. If you want to find out which drug made the most money for a company in 2015, it was a thing called Humira, which I would lay fairly long odds most of you have never heard of. Some of you might be studying it in your groups, I don't know. But it's not a drug that sort of is on everyone's lips. It's not a thing that you might think about if you thought, what is the biggest selling drug in the world? So what is this, and why is it the biggest selling drug? Now, bear in mind, biggest selling drug here is in terms of revenue. It's not how many boxes or how many doses of Humira was sold. It's which drug generated the most cash for pharmaceutical companies in 2015, which, from a company point of view, of course, is what they're most interested in. Humira is a monoclonal antibody. It actually acts on a protein called TNF-alpha, tumor necrosis factor alpha, and it's used by people who suffer from conditions like rheumatoid arthritis, which is an autoimmune condition where the body's own immune system starts attacking components in its own cells. I actually know someone who takes this, the wife of a friend of mine who developed very bad rheumatoid 2 or 3 years ago, and she's now on Humira, and it works incredibly well. She was able to live pretty much a normal life. So it's quite a breakthrough therapeutically, because before drugs like this came along, there wasn't really a good treatment for things like rheumatoid. So if you look, again, these are 2015 figures, but if you look at the top 10 revenue-generating drugs in the world in 2015, here they are. So Humira is the one that sits at the top. And if you look at the type of these drugs in this third column here, you'll see they're basically of two types, biologics and small molecules. Small molecules are the things that you might think of when we normally talk about drugs. You know, things like aspirin, relatively small compounds, probably made using straightforward standard organic chemistry. But biologics means proteins or large peptides. So things like insulin, for example, would be regarded as a biologic. So if you look at this list, one of the things you can see straight away is 7 out of the 10 top grossing drugs in the world are biological therapeutics. They're not small molecule drugs. You can see here the sort of income that they generate. These are millions of dollars per year. That's a lot of money. That's $14,000 million a year, $14 billion from the sale of that particular drug. And if you look at the trade names, the drug name down here, what you'll see is that most of the biologics, in fact all of them apart from insulin, which is number 6, which is treatment for diabetes, obviously, all of them end, and actually this one here as well, end with the suffix MAB, M-A-B. They're all monoclonal antibodies. So a substantial proportion of the drugs that make drug companies their money are monoclonals. And people have tracked this over the years, because monoclonals are very new drugs. They weren't around really 10 or 15 years ago. They're a new entry into the pharmaceutical market. And year by year, they've generated more income for the drug companies. 2013 was actually the first year when the income from the biologicals, the therapeutic biologicals, was greater than that you get from the small molecules. So small molecule drugs tend to be very cheap, and they're sold in extremely large amounts. The biologics tend to be very expensive, and they're shown that they're sold in smaller amounts. But from the drug company's point of view, they actually make them more money. So these are the things that we're talking about in the latter half of this course. You'll hear them called therapeutic biologicals, biotherapeutics, biopharmaceuticals, biologics. It all basically means the same thing. And what it means generically is proteins with some kind of therapeutic use. And these days, pretty much all of these are derived from organisms which have been manipulated genetically one way or another. So insulin in the old days, of course, was largely made from pigs, but these days it's pretty much all produced by either E. coli or engineered yeast or human cells. And note the distinction from the small molecule drugs. So these are large molecules, generally proteins. Small molecule drugs typically have molecular masses of 100 daltons, 200 daltons. These have got molecular masses of many thousands of daltons. And here's some examples of things that you'd class, generally speaking, as therapeutic biologicals. Cytokines, hormones, blood clotting factors, vaccines, antibodies all fall into this category of therapeutic biologicals. What we're going to be focusing on in my lectures and the lectures which are going to follow, which I'll tell you about, is the antibodies, because these are the really important ones from a therapeutic point of view. But I will say something today briefly about some of these other ones as well, just so that we don't totally neglect those. So what are we going to cover? We'll cover structure and function of antibodies, how they can be re-engineered, and this diagram will become clearer to you later on, how you can go from an antibody, which is basically one made by a mouse, to an antibody that recognises the same thing but is identical to a human antibody. How they're used both in diagnostics, which is a very important use of antibodies, and therapeutically, and importantly for this course, because this is a biotechnology course, how they're produced, how you actually make these things commercially speaking, because they have to be made in reasonably large amounts in order to be sold by the pharmaceutical companies. And you're also going to get some lectures comparing the two, actually in the context mostly of cancer. So how do these different sorts of molecules measure up when it comes to using them to treat cancer? A fairly important target, obviously, of the drugs industry. So these lectures are going to be by myself. Next 4 lectures, 2 today, 2 next week, and by Mike Tomlinson. Mike will go over some of the stuff that I've gone over, but from a different angle. Mike's much more of an expert, actually, in pharmacology and pharmaceuticals than I am. I know a bit more about the molecular biology side of things. So between us, we should give you a reasonably comprehensive survey on how these small therapeutic biologicals work. And then as far as protein production is concerned, so how in particular these molecules are produced, and we're talking here about mostly the antibodies, but everything that you learn in these lectures applies to other biotech products as well. How are these made by engineering bacteria, simple eukaryotes like yeast, and complex eukaryotes like mammalian cell cultures, which is shown along the bottom here. And that is going to be done by Dr. Huber, who you've met already. He ran the workshop for you last semester on primer design. And again, Mike will talk about production from mammalian cells. So by the end of all this, you should have some idea of what these molecules are, why they're important, how they're produced, and very importantly, how you decide what kind of organism you're going to use to make them. What are the factors that determine whether you're going to make these things in E. coli or in a yeast? And if you're going to use a yeast, which yeast are you going to use, or are you going to use mammalian cells? Because there's pros and cons to all those different routes of production. It's important that you're aware of that. You're then going to have a small set of lectures on stem cells. These don't really fit into the therapeutic biologicals area, but we're very keen you should learn something about them, so we've basically shoehorned them into this module. You'll learn a little bit about what stem cells actually are, different kinds of stem cells, and why they cause a great deal of interest from a clinical point of view. They haven't perhaps completely realised their potential yet, but there's a vast amount of research going on them, and it may well be in the next 5 or 10 years there'll be some really significant advances made there. That's going to be Laura O'Neill. She'll talk to you about the stem cells. And one other thing that we've shoehorned into this module is a workshop which I will run. This is just not a practical workshop. This is just learning about the technique and then looking at some data. And this is learning about flow cytometry and how you can use flow cytometry to separate different cell types, which can be useful both in terms of measuring the presence of those cell types, but also even, if necessary, purifying them. And we'll look at some examples of flow cytometry data and go through those in the workshop, so you get some idea about how that's used. It's quite likely, incidentally, that some of you will be doing flow cytometry in your research projects. So this will be an introduction to it for those of you that haven't come across this particular technique before. The assessment of this part of the module is basically a class test that we held in week 10, so quite a way off still. And that's going to be, I think, 4 short answer questions, and that'll be based on the material that I'm talking about and that Mike and Damon are going to cover, and also the flow cytometry workshop. There'll be a question in there on that, and that's worth 30% of the mark for this module overall. More detail about this will be posted on Canvas, obviously, as we get closer to the time of the test, so you'll have some idea about the sorts of questions that you'll have in that assessment. Okay, so let's go back to this list, the different kinds of proteins that we're referring to when we're talking about biological therapeutics. And these are all produced these days using recombinant DNA-based methods. So they're all produced by cloning the genes and expressing the proteins from those cloned genes in a suitable production organism. You'll hear much more about the choices of production organism from Mike and from Damon. I'm going to start off by saying something briefly about cytokines. I don't want to talk a lot about these. They are quite important medically, but they, as you saw from that previous list, they're not massive. Cytokines are cell signaling proteins. So they're proteins which are soluble, they circulate around the body, and they bind to receptors on particular kinds of cells, and those cells then respond by producing various different compounds. You might think that that definition overlaps with the definition of hormones. You're familiar with the concept of hormones, some of which are proteins or peptides, of course not all of them are, which again circulate around the body and affect different cells. There's a couple of differences though. When we're talking about cytokines, first of all, generally they're active at lower concentrations than hormones. Importantly, they're secreted by a large number of cells. They're secreted specifically by different cells which are part of the immune system, whereas hormones, of course, are secreted by specialized glands in the body, like the adrenal gland and the pituitary gland. So the site where hormones are produced is very precise within the body. Cytokines are produced from cells, particular cell types, that circulate all the way around the body. And very, very, very broadly speaking indeed, cytokines can have 2 kind of effects. They can either stimulate a particular component of the immune system that's responsible for inflammation, and you're all familiar with this. Anytime you've scratched yourself or cut yourself and you get that kind of red raised area around the scratch, which gets a bit warm and a bit puffy and a bit sore, that's a classic inflammatory response. So we divide cytokines crudely into 2 groups. They either cause that inflammation or they stimulate that inflammation. We call those pro-inflammatory cytokines, or they suppress it, and we call those pretty obviously anti-inflammatory cytokines. And inflammation, we'll say a little bit about later on. It's a very, very complex process, and the balance of different cytokines will influence the extent to which an inflammatory response actually happens. They've got lots of different functions in the body. For example, the interleukins are very important. Some of them stimulate the differentiation of T cells. We'll be talking a little bit about T cells later on today. Some of them can suppress B cells, so this is a pro-inflammatory, this is an anti-inflammatory response. There's the interferons that you may have heard about. There was a great deal of excitement about at one point. They've kind of gone off the boil a little bit now, but they're produced by the body during viral infection, and they help to activate the immune system. And there's plenty of other examples. They tend to be, for the most part, quite low molecular weight proteins, and they're often very simple in structure, monomers, dimers. So this is 2 cartoons of 2 typical simple cytokines, interleukin-2 on the left, interleukin-10 on the right. Incidentally, it occurred to me when I put this diagram in that it was possible that some of you perhaps hadn't done a lot of structural biology in your first degrees, and you might not be completely clear about what these sorts of diagrams are actually showing. So I thought I'd just take a sidestep for a couple of minutes to say something about when you see these kind of pictures, and you'll often see these in talks and seminars and see them in papers and so on, what does this actually represent? Obviously, these are proteins, and pretty obviously, both of these proteins are mostly alpha helical. So these red springy structures here, these are just cartoons showing the alpha helix. And the other parts in kind of bluey grey are unstructured in the sense that they're not either an alpha helix or beta sheet, although they do have a particular location in space. Now, what is very important to realise is that proteins don't look anything like this. This is not what a protein looks like. This is just a cartoon representation of a protein to make it easier for us to get a sense of what the protein structure is like. So if we've got an alpha helix, we draw this spiral, for example. But don't go away thinking that proteins look like springs joined up with pipe cleaners. They really, really don't. So I've got some other diagrams which are cartoons of one particular protein. It's a dimeric protein, just to give you a sense of these different representations that you will see. And it's important, all of these are called representations. They don't really, you can't see proteins, at least not with the naked eye. You can see them with powerful microscopes. But don't run away with the idea that they look anything like this. So here's an example of a similar cartoon. This is a different interleukin. You can't really tell from this anything other than the fact that it's got alpha helices in it. It's a little bit hard to get any other information. If you colour the two chains differently, which I've done here, you can see straight away this is a dimeric protein. So I've coloured one chain orange, one chain grey, and you can see there very, very neatly, I hope, that there's two chains intertwined around each other, but the structure of those two chains is basically the same. This kind of shows you why we use cartoons. This is a representation that shows a little stick for every single bond that's joining two atoms. The only things which have been taken off these are all the hydrogens. So there's no hydrogen atoms shown here. And this is getting to be a little bit more what a protein might actually look like. And you can't see the alpha helices in there. Okay, it's not at all obvious from a stick diagram like this that there's any alpha helices. But this diagram and this diagram are exactly the same protein in exactly the same representation. So that's why we draw these cartoons. It just helps us very quickly to get a sense of the structure of the protein. But as you can see here, if we actually were to draw in all the individual atoms and bonds, it would look very, very different. And it's really hard to spot anything here. There's an aromatic ring here. It's probably a phenylalanine or a tyrosine or something. But most of these things, just by looking at it, it gives you very little information at all. We can draw a diagram like this. This is what is called van der Waals radii. So these represent slightly more what you might think the protein looks like in space. These are now spheres that have been put around all the individual atoms, depending what the van der Waals radius of that particular atom may be. So now this protein, this molecule, is the same as that molecule. Okay, but now you get a much better sense of how much space those atoms actually physically take up in the protein molecule. And you can see that whereas the cartoons tend to give the impression that there's lots of empty space in the protein, that's absolutely not the case. So proteins are mostly not empty space, they're mostly filled with the amino acids. And we can kind of smooth these and do some moderately complicated chemistry to say how will the van der Waals radii act when there's other atoms nearby. And this is now getting a little bit more like, if you could physically see a protein, it would look a little bit more like this. Now, there's almost no information that the naked eye can see here in terms of the secondary structure. You really can't see that there are alpha helices there. But you start to see interesting things, like you can see there's some kind of channel here maybe, or maybe this is a binding pocket. So quite often when people are looking at structures, they'll look at all these different kinds of representations to try to get some sense of why does that protein have the structure that it has, and what can you learn about the function of that protein just by looking at the structure. And then you get even fancier ones like this, where you can see both the van der Waals radii, which is shown by this mesh, but also the cartoon of the alpha helical structure underneath the protein. So I just wanted to show you those figures just to kind of remind you that when we do show these sorts of diagrams like this, they really are just cartoons. They don't show you what the protein looks like in actual physical space. They just show the secondary structure. And you will see a lot of those in your reading. Okay, so back to cytokines specifically then. One of the things that cytokines initially people looked at with great excitement. They thought they'd be very useful clinically. One of the problems is that they act with lots of different components in the immune system. They have lots of different targets, lots of different cells that they work on. And in those actions, they have diverse effects. And this limits their clinical usefulness because there's loads of side effects of using cytokines. If you're trying to target one particular process by modulating cytokines, it isn't going to work because the action of cytokines is so complicated. All of them work through things called cytokine receptors, so that although they're small proteins, they still can't penetrate inside cells. So they work by binding to other proteins which are embedded in the cell membrane, and that binding causes those proteins to change conformation and then to transmit some signal to the inside of the cell. So clearly, because they have to bind a receptor in order to work, they can only work on those cells that express that particular receptor. If a cell doesn't have a receptor for a cytokine, it won't see that cytokine. The cytokine will only affect a cell that's expressing a receptor for that specific cytokine. And in fact, it's turned out that often the receptors are better targets than the cytokines themselves. Now, Humira, as it happens, is a TNF-alpha, is an antibody that targets specifically TNF-alpha, but there's another range of drugs that target the TNF-alpha receptor. So you can either block the cytokine by binding to the cytokine, or you can block its activity by binding to the receptor for that cytokine. Interferons, I've mentioned already, is an example of cytokines that have clinical use. There's lots of different kinds of interferons, interferon alpha, interferon gamma, interferon beta. Interferon beta 1A, 1B are one of the few treatments that we've got available against MS, multiple sclerosis. They're not cures at all. They slow the progression, but they certainly don't cure it. And they're glycoproteins, which means that they've got large sugar residues attached to their surface. And commercially, interferon beta 1A is made in mammalian cells, and 1B is made in E. coli. Then another example here, interferon alpha 2A and 2B, and this has been approved for treating a range of different cancers. So it's one of the one of the weapons in the arsenal that clinicians have against things like leukemia, some kind of leukemias. Kaposi's sarcoma, which becoming less common now, but was very common in the early days of the AIDS epidemic. Malignant melanoma, skin cancer, which is quite a problem, especially in countries where people sunbathe a lot. So these are just a few examples of cytokines which have got some kind of clinical use. Another example of therapeutic biologics, and these have been around in the market for quite a while, are the blood clotting factors. So blood clotting, you're all familiar with. This is the process that takes place if you cut yourself, or indeed if you have a bash and you bruise. A bruise is just internal bleeding under the skin. And the reason that doesn't keep on happening forever and ever and ever, and you therefore bleed to death, is that fairly rapidly a cascade of events takes place within the blood that finishes up with cleavage of fibrinogen to fibrin, which then becomes cross-linked and forms a clot, and that prevents the vessel from leaking any further. You may well have looked at this in your biochemistry and biology degrees. It's a complicated cascade of events where proteins are present in an inactive form, and they become cleaved locally to the active form, and that then cleaves another protein, and eventually you get a clot forming here. And the reason for this is kind of obvious. Blood clots are really, really bad news, except in the place where you want them. You don't want a blood clot in your brain. That's what causes a stroke. You don't want a blood clot in your heart. That can cause a heart attack. You don't particularly want blood clots anywhere in your body except for where the body's damaged in some way. So the whole process, the whole cascade process in blood clotting has evolved in order to only allow clots to form at the site of damage and not to occur anywhere else. But some people are unfortunate enough to lose some of to not express the genes or to have mutated genes for some of the key components of this cascade. Factor VIII is the classic one, so if you don't have factor VIII, if your body can't make enough factor VIII, you'll suffer from a condition known as haemophilia A, and that is potentially a fatal condition, because if you cut yourself, you don't clot, and even a minor cut, you could bleed to death. There's a slightly rarer form called haemophilia B, and that's due to a loss of factor IX. Now, haemophilia is bad not just in the case of a superficial cut, but also people can get bruising, joints, for example, during growth. You can get bleeding inside the joints, and that doesn't stop, and then the joints don't develop properly. So there's all sorts of reasons why haemophilia needs to be treated. And for many years, factor VIII has been available commercially. It's essentially a lifeline to people who suffer from haemophilia A. Originally purified very, very expensively and very difficultly from blood, these days produced as a recombinant protein from a simple organism. In fact, yeah, most Advate, which is the commonest form of factor VIII, is produced from Chinese hamster ovary cells, CHO cells, which are a particular common kind of tissue culture cell. Okay, so I'm not going to say anything more now about cytokines or about blood clotting factors. You just need to be aware of them. It's possible some of you might do some cytokine work in your projects, because there's quite a bit that goes on up in the med school, so some of you may be working on those later on. Vaccines have been around for quite a while. They're probably, after clean drinking water, which is the best thing you can do to improve the health of the planet, vaccination is probably the next best, and it has had huge effects on health, initially in the Western world, but now increasingly throughout the developed world as well. Now, in order to understand how vaccines work, we need to discuss the immune system, and this is going to be a huge sidestep now, because I want to talk quite a lot about the immune system and the components of the immune system, both so that we can understand vaccines, but more importantly because, as I've said, some of these components of the immune system are very, very important as drugs. So I'm going to basically talk for the rest of today, and actually the lectures next week as well, about the immune system, and quite specifically about antibodies. This isn't a course on immunology, so it's not a complete story. Those of you that have done a lot of immunology already, this will probably be going over old ground for you, but for some of you it may be completely new. I want to point out one particularly good book. This is an absolute classic textbook called Janeway's Immunobiology, which is now written by, authored by somebody called Murphy. And actually most of the diagrams that I've used in the lectures are taken from that book. So if you want to go and read more about that, or the things I say that you don't understand, it's a really good book to look at. There's lots of copies up in the library because the medics use this all the time. Okay, so the immune system. We're talking here just about the human immune system. Not going to think about immune systems in different organisms. And you have within you, as most of you probably know, two different aspects to your immunity. There's your innate immune system, and there's your adaptive or your acquired immune system. And these are really quite different, although as we'll see, they do overlap to a certain extent. And the innate immune system itself can be divided into sort of two components. There's what's called the immediate innate immune response, and then there's an early induced response. And we'll look at all of these in a lot of detail. The immediate response basically happens straight away if you get some kind of foreign organism into your body. The first line of defense, once the physical defenses are broken down, like the skin and the gut and so forth, the innate immune system is the first line of defense, happens straight away. Then the induced immune system takes a little bit longer because it requires some recognition to take place. And then the adaptive immune response, which is the one that we often think about, and this is the one that you're targeting with most vaccines, takes a lot longer. But the great thing about the adaptive immune response is it remembers. And so the adaptive immune response forms the basis for immunological memory. And this is why many conditions you don't get twice. So once you've had a condition, you don't get it again because your immune response has learned how to respond to it, and that's therefore also the basis for vaccination. So I'll say something about the innate immune system, and then we'll move on to the adaptive immune system, because the adaptive immune system is where the antibodies come in. So innate immunity. Innate immunity refers pretty obviously to an immune system that acts fast, doesn't require expansion of B cells, doesn't involve antibodies, doesn't involve T cells. They're all part of the adaptive system. And the innate immune system, as I've said, can basically be broken down into two components. And the first one is what we call immediate innate immunity. And the components of the immediate innate immune system are always present. So these are always circulating in your blood or present in your tears. And this just consists of a series of different compounds. Many of them, for example, are antibacterial, and they're just operating all the time. So your tears, for example, contain lysozyme, which digests bacterial cell walls. Your blood contains antibacterial peptides. These are small molecules called defensins, which punch holes in bacterial cell membranes. There's also something called complement that I'll say a bit more about later on that acts in different ways, but one of the ways it acts, again, is by punching holes in cell membranes. So you, of course, don't realize this because your innate immune system works, but you're constantly being exposed to a barrage of different bacteria, and most of these don't get past these early stages. It is interesting, incidentally, to look at immune systems in other organisms. So amphibians, for example, don't have an adaptive immune system, but they've got a very, very effective innate immune system. And in particular, they produce lots of antibacterial peptides. And there's a great deal of interest in these as alternatives to antibiotics, getting hold of these peptides and then using them as treatments for infections. So even the simplest forms of the immune system are studied in a lot of detail for people who are trying to find novel ways of treating infections. Then we've got the induced innate response, which is a little bit slower because it does require the body to recognize that there's some kind of infectious organism present. So it's not something that's present all the time. It requires a recognition step. And in the induced innate response, the body contains a whole set of receptors that are able to recognize things that are found on pathogens but aren't found in the body. The classic example of this is lipopolysaccharide. So lipopolysaccharide is an important component of the outer cell membrane of many bacteria. We don't make it ourselves. We don't contain any LPS. So if any LPS comes into our body knows that's bad news. It's not something that we want to have around. It's not home, it's foreign. So by recognizing signals which are produced by pathogens, the body can defend itself against attack from those pathogens. Now, pathogens, of course, are always evolving, and so what pathogens often try and do is to fool the innate immune system by not possessing those signals or by producing signals that look more like host than they do look like foreign. What does this recognition? So the recognition is done by a whole range of different cells, but often these cells fall into the general class of what are called phagocytes. Phagocytes, again, is one of these generic terms. There's lots of different cells which are phagocytes, but broadly speaking, a phagocyte is a cell that can recognize and bind to and take up and eventually break down foreign bodies like bacteria or fungal cells. You'll hear people talking about professional phagocytes and non-professional phagocytes. Professional phagocytes are very specifically there to recognize pathogens like bacteria. Non-professional phagocytes can also engulf substances, but they're not specifically part of the innate immune system in the same way as the professional phagocytes are. So what happens when a phagocyte recognizes a compound or a structure which it knows is not human, it knows comes from a bacterial cell or a fungal cell, it will bind to that cell and eventually it will take that cell up into the body, into the cell, and it's then broken down in a structure called a phagolysosome. I think I've got a diagram of that here. Yeah, okay, so this is an example. This is a macrophage, which is one particular kind of phagocytic cell. Without going into the details, you'll see that this macrophage has got lots of different receptor molecules in its outer membrane. So all these little coloured diagrams here are different receptors that recognise different sorts of structures that are found on foreign cells, including bacterial cells. This receptor here called, yeast cells, Dectin-1 can recognise yeast. This is a complement receptor. I'll tell you more about complement in just a moment. If these receptors are triggered, if they recognise a structure which they can see as foreign, what happens is that eventually the cell invaginates around that structure, takes it into the cell in what's called a phagosome, and that's then fused with the lysosome. The lysosome contains lots of destructive enzymes which break down the invading body. So this is the fused entity is called a phagolysosome. And those receptors are then recycled back to the cell surface where they can do their job all over again. Now, the general name given to structures on pathogens which are recognized by the induced innate system is pathogen-associated molecular patterns, or PAMPs for short. So you'll hear people talking about PAMPs. What they're talking about is any kind of structure which the innate immune system has evolved to recognize and to deal with. And generically, the receptors that recognize these structures, these PAMPs, are referred to as toll-like receptors, or TLRs. And you may come across these terms quite a lot in your reading or in other lectures or seminars. So PAMPs, basically any kind of structure that a pathogen expresses that can be recognized by the body, and the thing that does the recognition is the TLR, or the toll-like receptor. And there's quite a lot of these. We don't know what they all recognize, actually, but here's some examples of the main toll-like receptors. TLR11 actually isn't present in human, it's only present in man. So, for example, flagellin. So bacteria swim around using flagella. The flagella are made of a particular protein called flagellin. And we have in our gut receptors that can recognize the flagellin. So that's TLR5, toll-like receptor number 5. Some receptors are adapted to recognize RNA from viruses, so double-stranded or single-stranded RNA. Up here we've got receptors which are interesting. These recognize structures called lipomanams, which are found in the cell walls of mycobacteria, so the organisms that cause tuberculosis. And what you can see is that these different receptors are distributed in different cell types. These are all either phagocytes of one sort or another, or in the case of natural killer cells, NK cells. These are cells that can secrete material that destroys the stuff that they're bound to. I talked earlier on about LPS, which is found in the outer membrane of gram-negative bacteria. There's also a compound called lipoteoic acid, which is found in the single membrane of gram-positive bacteria, and these are recognized by these TLR4 receptors, which are found, as we've seen, mostly on macrophages, but also these other cell types here, dendritic cells, mast cells, and so on. So you're probably beginning to get the picture. This is pretty complicated, and it's not essential for you to know all these details. What you need to know is specific examples. So you should be able to say, here's an example of a PAMP, this is the kind of receptor that it binds to, and this is the kind of cell type that that receptor is found on. I'm not going to ask you in any exam question to reproduce that table. You don't need to know that level of detail. So any kind of induced response requires a number of steps, and the first step is always recognition. In order for the response to take place, cells have to realize that there's something around that shouldn't be there. So the first step is the recognition of the PAMPs, the molecular patterns, by these toll-like receptors. The cells with those toll-like receptors respond to that recognition by producing pro-inflammatory cytokines that we've already talked about. So the inflammatory response, which remember is a very quick response, it happens within minutes of something like a scratch or a sting, same sort of thing, happens because there's entry into your body of things which are recognized by cells as being foreign. Those cells then produce cytokines. Cytokines are of the type that induce the inflammatory response. You get inflammation around the area where the damage has taken place. inflammation isn't only caused by foreign bodies, it can also be caused by tissue damage as well. So there's various different things superimposed here. And the inflammatory response, we're not going to talk about, you don't need to know the full details of it. It's a complicated, actually fairly long-term response that then attracts other agents of the immune system, basically so that we stay healthy. So if we don't have this, you're much more likely to become infected and develop diseases because, again, this is a really important barrier against the egress of any kind of, or the ingress rather, of any kind of infectious organism. Okay, that's enough about the innate response. We'll come back to look at some components of that later on, like complement in particular, because complement is kind of shared between the innate and the adaptive response. So the second component of the immune system, and this is probably the one that you're a little bit more familiar with, is what's called adaptive immunity. And this is much more specific. So rather than recognizing general molecules like LPS or teichoic acid or the other examples I gave you, which are broadly present on pathogens, the adaptive response recognizes very precise patterns within specific molecules. And we'll look in quite some detail at how that happens, both because it's very interesting, but also because we need to understand how that happens in order to be able to engineer antibodies that are going to be used therapeutically. So we'll go into that in quite a lot of detail later on. The consequence of that recognition is what I'm going to talk about next. And the consequence of that recognition is the expansion of two populations in the body in particular, and these are the populations of B cells and T cells. And again, I'll tell you a lot more about those in just a moment. It's slower than the innate response. It takes hours to days in order for this to take place. And the reason that it's good that we've got both systems is because when you're infected, the body can't wait for days and days for a response to take place. It needs to deal with things in the short term. But by having the adaptive system, you make the whole response of the body much more specific. Now, the important thing about the adaptive immune response, as I've said before, is it leads to immunological memory. Once the body's produced an adaptive immune response to something, it changes the circulating populations of cells in such a way that if that entity reinfects, the body is going to respond much more quickly. And that's why once you've had a vaccination against something, as long as that adaptive memory remains within the body, it does decay slowly over time, but as long as you've got that adaptive immunity, you're then immune or largely immune to infection by that particular condition. So let me just, again, you don't need to learn all the details on this diagram, but we'll be talking about different sorts of cell types within the immune system. And this shows the way in which these cells are produced. We've been talking a lot about macrophages and other phagocytic cells like dendritic cells. And these are derived from progenitor cells called granulocytes. Most of these are cells which are circulating in the blood, so they're examples of the white blood cells, some of the white blood cells. But they don't have to be found in the blood. Some of these cells are crawling around your epithelial surfaces. So the inside of your gut, for example, has got some of these cells moving around dealing with pathogens. And then you've got the lymphoid lineage, and these are the cells which are classed as lymphocytes, which of course are also circulating in the blood in most cases. And these divided into two groups, three groups, the B cells, the T cells, and the NK, or the natural killer cells, that I've said a bit about already. The B cells are the ones that produce the antibodies that we're going to be talking mostly about. T cells have a different role, and I will say something briefly about natural killer cells as well. So we've got two crucial cell types involved in the adaptive immune response. There's what's called humoral immunity, and this is what's mediated by the B cells, and the B cells are important because they're the source of antibodies. And we've got cell-mediated immunity, and this is mediated by the T cells. They're both really important. They're very interlinked, and you can't talk about one without talking about the other, because the T cells are involved in the B cell response, as you'll see. But I'm really mostly going to focus on humoral immunity because, again, the thing I really want to talk about in detail is the structure of the antibodies and where they come from, how they're produced, how the body learns to recognize particular structures with enormous specificity. T cells are used therapeutically. There are T cell therapies out there. You may hear about those. They're less easy to work with than antibodies because for a T cell to be active, you require the whole cell to be there. For the antibodies, you only require the antibody. So that's why antibodies are more attractive as a therapeutic agent. So this is what antibodies look like. We'll keep on going back to these structures. The simplest way that you can show them is here. This is a particular kind of antibody called IgG. There's different classes of antibody. They contain typically 4 protein chains. 2 of these are referred to as the heavy chain, 2 as the light chain because of their molecular weight. These chains are all cross-linked to each other by disulfide bonds, which are shown in yellow here. And they also contain a fair amount of disulfide bonding within the chains themselves. And this is a useful diagram. There's a lot of detail here, but there are 2 really important parts of the antibody. There's this top bit, the head of the antibody. This is sometimes called the Fab region, and this is the part of the antibody that does the recognition, so that recognizes foreign structures. And the things that they recognize are called antigens. So anything recognized by an antibody is by definition an antigen. And we'll get into the details of what's going on in these hypervariable regions later on. And then you've got the tail of the antibody, which is just this end of the heavy chain, so the light chains aren't involved in this. And this is important for the biological activity of the antibody. It doesn't do anything with the recognition, but the question is what happens after recognition has taken place, and that function, as you'll see, is encapsulated in this part of the antibody, the FC region. So you've got the FAB region, which basically does the recognizing, and the FC region, which is the active part in terms of biological function. Don't know if you've come across a company called the Binding Site. The Binding Site is a spin-out from this university that produces extremely large amounts of antibody, actually for diagnostic purposes. Very, very successful company. They were originally based in a kind of a garage fairly close to here. They now occupy a very spectacular site up by the Five Ways roundabout. And if you look at the outside of the building, you'll notice they've got a big picture of an antibody in the window, because that's essentially what they produce. Now, 999 out of 1,000 people will walk past that building without having any idea why it's got that image in the glass windows. But next time you're up in Five Ways, if you walk down that way, you can walk past that headquarters. And actually all the production's done there as well. Remarkable place, and that's what their wealth is founded on, basically. Right, so antibodies, also called immunoglobulins, are large, complicated proteins. They come in various different forms. Their key function, or the thing that makes them really remarkable and unique, is that they can recognize and bind very, very tightly to a whole range of other structures. And these other structures are called antigens. And this is what makes them so powerful. They're very powerful as reagents in the lab. They're very useful as diagnostics. And as we'll see, and as you've seen a little bit already, they're also very powerful as therapeutics. They're highly, highly specific. And we'll see later on how this specificity works. But essentially, they have surfaces which are complementary to, or fit into very nicely, like a hand into a glove, to other structures that they have to recognize. Now, there's all sorts of interesting questions about antibodies that we're going to address in the next hour or so and next week. Like, how come they don't recognize us? How come they don't recognize all the structures in our own bodies? And given that you can find antibodies that can recognize tens of thousands of different structures, where does all this variability come from? How come the genome, which is finite in size, is able to encode all these different proteins that differ in their ability to recognize different structures? So if you don't know that already, which some of you may do, you'll learn more about it during the course of the next 2 or 3 lectures. And then the question is, what do they actually do? Okay, so they do this recognition, that's really cool, but having recognized things, what happens next? Why is that important in the action of the immune system? So we'll take a look at that as well. I think actually this is probably quite a good place to take a break. I've been yattering on for 50 minutes or so, so let's take a 10-minute break at this point. I will pause the recording to